Hi, my name is Minhaj Bhuyan, and I'm joined with Jaden Lee and Sabrina Lorza, and today we're going to be talking about the structure-function relationship of musculoskeletal microstructure.
Skeletal muscle is extremely vital to bodily functions such as breathing and can be applicable to clinical cases such as pressure ulcers. Investigating skeletal muscle also provides better insight of the biomechanics world. Our hypothesis is threefold. First, with the hypothesis that biaxially stretched tissue will be significantly stiffer than uniaxially stretched tissue. Secondly, we hypothesize that there is a direct correlation between intramuscular pressure and compressive loads. Lastly, we hypothesize that tension-compression asymmetry can be modeled via a von Mises bivariate fiber distribution.
In order to study and test the hypotheses presented, three methods of experimentation were chosen. To test stiffness under stretch, multiaxial and uniaxial tensile testing was conducted. To test intramuscular fluid pressure, uniaxial compression testing was conducted. And finally, to test tension-compression asymmetry, multi-scale finite element modeling was conducted. For the experimental portion involving muscle tissue, fresh porcine hind limbs were obtained from a local farm. The tissue was promptly dissected in order to avoid effects of rigor mortis into two different sample types. For the tensile tests, cruciform-shaped samples were prepared, and for the compression tests, cube-shaped samples were prepared. The tissue was loaded into a biaxial testing machine and either stretched or compressed, and the raw force data from the biax was collected. In addition, a digital image correlation system was used to capture strain data for the tensile tests, and a fiber optic sensor was used to obtain pressure data for the compression tests. All data was processed and analyzed in custom-written MATLAB scripts.
Computational simulations were created to generate theoretical stress stretch curves for anisotropic asymmetric muscle tissue. First, muscle tissue was virtually generated via Gibbon code, an open-source MATLAB image processing plugin. Then, biaxial boundary conditions were applied to the geometry in the finite element package FEBio. Finally, a Monte Carlo simulation was used to find theoretical parameters that would generate stress stretch curves that would match experimental data via MATLAB. A relationship in the experimental data can be observed between microstructure and mechanical function. In Figure 6, fibers undergoing uniaxial compression yield higher fluid pressures than solid stresses and can be observed to have a nearly direct relationship with one another. In Figure 7, muscle fibers undergoing longitudinal tensile stress become increasingly stiff with increasing strain, while fibers in transverse tensile stress increase in stiffness at a lower rate. The finite element simulations show that the bivariate von Mises parameter gamma causes the largest amount of variance in the stress stretch data in Figure 8, while other parameters yield relatively low variances in Figure 9. In conclusion, fiber orientation is crucial to muscle stiffness and can help advance our understanding of muscle force transmission. Future work will focus on collecting more multiaxial data to better validate and improve finite element models of musculoskeletal microstructure. From the MMOT, thank you for listening.